Every human cell holds about 2 meters of DNA. Yet that entire length has to fit inside a nucleus measuring just 10 micrometers across. That is like packing a string the length of a football field into a marble without tying a single knot. A biological system solves this problem through a highly organized sequential folding sequence called hierarchical packaging. To achieve this compaction, the cell relies on a composite material called chromatin. Chromatin is built by combining raw DNA with specific structural proteins, primarily histones, along with various non-histone proteins. If the cell merely shoved the raw DNA inside the nucleus, the resulting tangle would permanently block access to the genes. The structural proteins ensure the physical compression is tight enough to fit, but ordered enough that the genetic code can be unspooled, read, and accurately copied for cell division. This diagram shows the exact sequence of that hierarchical packaging. Starting at the top, the double-stranded DNA wraps exactly 1.65 times around a core of 8 histone proteins, specifically 2 copies each of H2A, H2B, H3, and H4. This forms a nucleosome, creating a pattern that looks like beads on a string. To lock this initial wrap in place, a single H1 linker histone attaches to the outside of the core. The addition of this linker protein creates a structure called a chromatosome. The chain of chromatosomes then begins coiling inward on itself, forming a dense 30 nanometer wide structure known as a solenoid. This solenoid fiber is then gathered into 300 nanometer loops, anchored to non-histone scaffolding matrices. These loops compress further into a 250 nanometer fiber, eventually condensing into the highly dense 1400 nanometer metaphase chromosome you see during cell division. By moving through these strict geometric stages, a cell successfully compresses its physical data by a factor of 10,000. It guarantees the entire length of material can be distributed cleanly into new cells without getting tangled. Once fully compressed for cell division, the metaphase chromosome takes its final shape. At this stage, the structure consists of 2 identical, parallel halves side by side. We call these sister chromatids, representing the parent DNA and its exact replicated copy. The chromatids are bound together at a primary constriction point called the centromere. It is crucial to distinguish this specific DNA region from the kinetochore. The kinetochore is a separate complex of protein disks anchored to the outside of the centromere, which serves as the physical attachment point for spindle fibers. At the extreme tips of the chromatid arms are the telomeres. These highly stable, repetitive DNA sequences cap the ends of the chromosome, preventing the genomic data from fraying or mistakenly fusing with other chromosomes. On certain chromosomes, you will also find a secondary constriction point, which acts as the nucleolus organizer region. This pinch physically separates a small bulb-like segment of DNA at the very end, known as the satellite. A chromosome is an engineered biological machine, equipped with mechanical attachment points for movement and protective caps to maintain structural integrity. This illustration demonstrates how the exact position of the centromere dictates the physical classification of the chromosome. The centromere divides the structure into a shorter top arm, labeled P, and a longer bottom arm, labeled Q. By evaluating the ratio between the P and Q arms, we get 4 distinct morphological types. Looking at the 2 shapes on the right, a metacentric chromosome has its centromere positioned directly in the middle, creating equal P and Q arms. If the centromere is shifted slightly off-center, resulting in clearly unequal arms, it is classified as submetacentric. Moving to the left, an acrocentric chromosome has a centromere pushed so far toward one end that the P arm is exceptionally short, often leaving just a satellite structure. Finally, in a telecentric chromosome, the centromere is located exactly at the terminal end, meaning there is effectively no P arm at all. Let's test your visual recognition. Look closely at this chromosome shape. The centromere is positioned extremely close to the top end, yielding a tiny P arm and a long Q arm, but it is not completely at the tip. Which of the 4 types is this? This is an acrocentric chromosome. Geneticists rely on these specific arm ratios and morphological shapes to map karyotypes. By visually identifying any structural deviations from these standard forms, they can accurately diagnose chromosomal mutations and genetic abnormalities. The primary biological purpose of chromosomes is the stable storage and physical transmission of heredity. They are the physical vessels that ensure genetic data transfers intact from one generation of cells to the next. They also control gene expression through physical density. Loosely packed regions of chromatin, called euchromatin, remain physically open and actively transcribe genes. Tightly condensed areas, known as heterochromatin, remain locked and inactive. During cell division, the mechanical structures we identified, specifically the centromere and the kinetochore complex, provide the physical anchor points necessary to pull the sister chromatids apart, guaranteeing an equal distribution of DNA. To recap, packaging begins with DNA wrapping into nucleosomes, which coil into solenoids and eventually loop into dense chromatids. A fully formed chromosome features a central centromere, kinetochore attachment points, and protective telomeres. Depending on the centromere's position, it is classified as metacentric, submetacentric, acrocentric, or telecentric. The eukaryotic chromosome is a feat of biological data engineering. It packs an enormous amount of genetic code safely inside a microscopic space, while maintaining perfectly ordered access to build, operate, and replicate the organism.